we're currently still retooling FAMRater for our own inputs, thresholds, and algorithms. FAMRater was initially, it utilizes a protein blast more than anything, and we are utilizing a nucleotide blast because we are trying to make DNA probes. And so we've got to change that throughout the program and other things that will aim it more specifically to our purposes. Also, we created a small test database. The Mycobacterium phage draft database is enormous. As I told you, it has like 34,000 genes in it, so that's a lot of information. And when debugging a program, it takes forever. It would probably take 2 days to run. So we made a smaller database that would be representative of it with the following families in it, so that we could debug the program. And then once we had it at a good level there, move on to the actual database. And lastly, and most importantly, we would need to validate those thresholds shown here in green, because that tells you whether or not it should be a new subfamily or not, and this is absolutely key when making the probes so that the consensus sequence represents them accurately. Okay, so speaking to the interdisciplinary nature of this project, I'm going to take sort of a left turn. The possum. There's a group of engineering students here at JMU that are building an arrayer, which is the thing that would print those 10,000 tiny little spots on that slide. And they are doing synthesis of the oligos directly onto the slide, so we'll be printing base by base by base. So it's incredibly impressive that students here at JMU are going to make this. They're almost done, and it lives in the HHS basement. So in order to help them test their printer, we wanted to see if they could print uniformly sized droplets, which is really important. We wanted to visualize these results, so we utilized the Psi 3 and Psi 5 fluorescent dyes that I mentioned earlier. And we needed to optimize the dye concentration for the test dye solution so that the bright spots were not too bright. In order to do so, this is our first trial, one of concentrations. The concentrations are shown above. And this crazy looking map is where we hand spotted each concentration onto the slide. We put them in this interesting pattern so that the results would not be an effect of a bias from the slide reader or something about the location on the slide itself, so that we would know they would only reflect the concentration itself. We did a trial on a glass slide and an aminosilane slide, and we used 50% DMSO as our spotting agent. We learned a whole lot from this. First of all, as you can see, the dye was way too concentrated. These spots are white. If you're actually doing a microarray experiment, that's useless to you. We also learned that the DMSO takes way too long to dry. Isaac and I probably sat in the lab for 2 hours waiting for it to dry, and it did not make any change. So we had to come back the next day. And obviously, this is not useful for the purposes of the possum. And we also found that the use of the aminosilane slide resulted in more uniform droplets. And that is the kind of slide that the possum will use, so that was good to know. So for our trial 2 concentrations, we used the following concentrations, and we changed our spotting agent to 1 times SSC. And this trial went a lot better. The optimal concentration was found to be a ratio of 1 times 10 to the negative 8 Psi 5. The 1 times SSC dried in approximately 15 minutes, so this was much more useful for our purposes. And we prepared the dye solution for the possum according to these results. Secondly, in order to test the possum, we prepared an oligo solution. We wanted to see if the possum could print previously synthesized oligonucleotides on a slide that would be used for successful hybridization. In order to do so, I chose oligos made from the George phage genome. And just so you know, oligonucleotides are just simply short strands of nucleic acids, that are usually less than 50 bases in length. I used Wiki and chose phage terminase and exonuclease to make the probes. And we gave the possum both of those solutions, and we will hopefully get those test results before the end of the semester. So in the future, hopefully by the end of the semester, maybe next year, we would like to do a potential macro array. Isaac and I found in the lab that it's not too hard to spot your own array. Obviously, it's not the neatest thing in the world, but it's actually kind of useful. And instead of having the possum print every single probe onto the slide, we could simply choose 1 or 2 and print or hand spot them ourselves and run a hybridization and see if they work. This is also possibly a really cool learning tool if a school does not have access to an arrayer. We want to use the possum to obviously print those probes that our program makes.